Okay, so we're going to lump together translation and post-translational gene regulation. Um, so we're out here in the cytoplasm. We didn't talk about export from the nucleus. There's a little bit of regulation there, but we did talk about whether the mRNA stays intact or not. But now we're going to talk about turning it into a protein. There are actually very few examples of regulation of translation itself. This tends to be controlled by the RNA mainly. But there are some examples of what we call post-translational modification. We can look at like the ribosome. The ribosome can get modified, and when this elongation factor is phosphorylated, the ribosome is unable to translate. When it's not phosphorylated, it's able to translate. This would be an example of translational control. This is very, very rare, and there's not a lot of great examples of this kind of control. In my time in science, over at least 10 years here, I've seen maybe one really good example where somebody was saying that there was translational control occurring. That's very, very rare, right? But what does happen a lot is what we call post-translational modification. It's really a lot more common. So we made our protein and it folded, then we can modify it in some way, adding additional molecules that might impact the protein's function. So one of the things that can happen, right, just like with mRNAs, is does your protein get degraded? If your protein gets degraded, that's gene regulation. That's turning the protein off basically by chopping it up. And an important post-translational modification that leads to protein degradation is called ubiquitination. Ubiquitin molecules are these little molecules that can be attached to proteins when they need to be degraded. When you get a chain of ubiquitin molecules building up, it's basically a red flag that calls out to this complex called the proteasome. A proteasome finds a ubiquitinated protein and breaks it down into its constituent amino acids. So it basically recycles the protein here. So ubiquitination is like the kiss of death for proteins. If it happens, they're signaled for destruction and recycling. Now, ubiquitination is one of many different types of possible modifications that could happen. We've talked about some of these. We talked about phosphorylation, where you add a phosphate to a protein. There are specific amino acids that can get phosphorylated. This can act like a signal that switches on or off the protein. It really depends on the protein, whether it's on or off. It's context specific. Glycosylation, you could add a sugar molecule to the protein. Ubiquitination, as I said, is a mark that targets proteins for degradation. There's one called sumylation, where you add a sumo, a small ubiquitin-like modifier, to it, and it can act like a trigger. Disulfide bond formation, where the two cysteine residues form a bond together. This one's actually really important in protein folding. There's a postdoc in a lab I worked in as an undergraduate who spent a lot of time trying to express a protein and then get it to fold into the correct conformation, the correct shape, because of these disulfide bridges. These disulfide bonds are also important if you have curly hair. When you apply a straightening iron to curly hair, it actually breaks these disulfide bonds in the hair proteins and leads to straight hair, at least temporarily. Acetylation, that's adding an acetyl group to it. Lipidation, putting a lipid molecule on there. Methylation puts a methyl group on. And hydroxylation puts a hydroxyl group on there. These can all impact the kind of activity of the protein, but these are all very context specific. So what they do depends on what the protein is, where it is, and many other factors. All right, so we said that direct control of translation machinery is very, very rare. More often, we're seeing proteins modified in a post-translational way. So after translation, we add things to them, like ubiquitin. That was a molecule we added that scheduled a protein for destruction. Or phosphorylation, which can act like an on-off switch. So these post-translational modifications, much more common, really kind of more difficult to deal with as well, because we don't have good ways of visualizing what's happening to a protein. We have a lot of good ways of looking at DNA. We have a lot of great ways of looking at RNA. Looking at proteins is much harder. if you're interested in looking at proteins, you have to know about a technique called mass spectrometry, which is very, very tightly linked to math and chemistry. So biochemists are very good at looking at proteins. Okay, so we've covered our regulation here, and in the final section, we'll talk about what happens when a gene expression regulation kind of breaks down and one of the potential consequences of that.